Welcome back to the deep dive. If you've ever dealt with chronic inflammation, especially allergic airway inflammation like asthma, you know the frustration.
It's relentless.
For decades, the strategy has been to just block the inflammatory messages, the cytokines like IL-4 and IL-13. But what if we've been looking at the wrong part of the battle?
What if it's not about the messages at all?
Exactly. What if the key to stopping these persistent drivers of asthma wasn't about blocking the signaling, but instead targeting the survival mechanisms, the literal eating habits of the cells causing all the chaos?
That is the fascinating premise of today's deep dive. We're focusing on the two main cell types that drive asthma pathology: type 2 innate lymphoid cells, or ILC2s, and T helper type 2 cells.
ILC2 cells, right?
These are the immune cells that, flood the airways with those classic inflammatory cytokines. And we're basing this dive on some groundbreaking research by Wing Johns and colleagues.
And our mission today is to really understand this metabolic superpower these cells get. What is the core insight the researchers found? I mean, how do these pathogenic ILC2s manage to expand and survive in such a harsh environment?
The central finding is that these pathogenic cells, the ILC2s and TH2s, they become addicted to a specific powerful antioxidant defense system.
Addicted.
Yeah, completely. They're essentially living on the edge of destruction because of their high fat intake, and they must constantly activate the system, specifically relying on two enzymes, GPX4 and TXNRD1, to prevent a type of cell death called ferroptosis.
So their very addiction to staying alive creates a precise, targetable vulnerability. That is a game changer.
It really is.
Okay, let's unpack this step by step. Let's start with a surprising nutrient that these inflammatory cells absolutely rely on to survive this allergic firestorm.
Well, first we have to remember that immune cells in inflamed tissues are often operating in really tough environments. Nutrients are scarce, conditions are stressful, and while many cells can just rely on glucose, these allergen-activated ILC2s show tremendous metabolic flexibility. They ramp up their ability to use glucose, fatty acids, amino acids, you name it.
But that adaptation comes with a catch, a critical dependency.
Exactly. And it turned out not to be the usual suspects, like glucose for quick energy.
So what was it?
The researchers did these long-term survival studies, about 72 hours, on ILC2s from challenged lungs, and they tested different fuels, supplying just glucose or just fatty acids. It failed. The cells just couldn't multiply.
But when they added amino acids,
the cells thrived. So they tested all 20 amino acids one by one, and the results were, well, dramatic. Only one, cystine, was absolutely required for ILC2 survival and proliferation. Remove cystine, the cells die.
Cystine, not a common name in, everyday metabolic discussions.
Yeah.
But it's the essential building block for their survival system. Why is it so crucial?
Cystine is the raw material for the cell's main internal defense shield, something called glutathione, or GSH. Think of GSH as the ultimate detoxifier, like the cell's internal rust-proofing agent. Cystine gets taken up, it's quickly reduced to cystine, and then it's used to synthesize this GSH.
And what does this glutathione do specifically?
It's the essential fuel used by this master anti-ferroptotic enzyme, GPX4. Imagine GPX4 as a dedicated waste management crew.
Okay, I like that.
Its job is to neutralize toxic oxidized lipids, that cellular rust, that builds up when the cell processes a lot of fat. And GPX4 needs a steady supply of GSH to do its job.
And the research proved this connection.
Oh, absolutely. When they starved the ILC2s of cystine, their numbers just plummeted. But if the researchers simply added supplemental ready-made GSH, the cells were completely rescued, proliferation was restored.
So cystine is purely critical for maintaining this redox balance.
Yes.
This sounds like a perfect target then. Just stop the cells from getting the cystine. But here's the next surprising wrinkle from the source material. These pathogenic ILC2s don't even use the standard front door to get their fuel.
Correct. The typical cystine importer for immune cells is called system XC-. But the pathogenic ILC2s, they show reduced expression of this transporter. They've swapped their delivery system.
For what?
They upregulate and rely heavily on a different transporter called ASCT2. This one usually handles other amino acids, but it also efficiently imports cystine.
Why would they switch? Does that make them more efficient, or is it just a sign that their whole metabolism is changing?
It seems to be a key adaptation for survival in that inflamed tissue. And critically, this swap creates a specific therapeutic advantage for us.
How so?
The researchers tested it. They used an inhibitor called restrain to block the common system XC- transporter, and nothing. It had virtually no effect on the ILC2s.
But when they blocked ASCT2?
When they used the inhibitor V9302 to block that specialized ASCT2 transporter, ILC2 survival just dropped, and signs of dangerous lipid peroxidation immediately skyrocketed.
So the normal immune cell detox pathway is, well, it's largely irrelevant here.
Pretty much. The pathogenic ILC2s have built this custom survival system, fueled by cystine, delivered by ASCT2, and protected by GSH.
And that dependency is the first layer of vulnerability we can exploit. Okay, so before we move on, let's quickly summarize the metabolic fuel they need.
Right. Quick summary, part 1, the fuel switch. Allergen-activated ILC2s are critically dependent on cystine for survival, which they convert into glutathione, GSH. This GSH is their essential defense shield. And to get this fuel, these pathogenic cells switch to using the specialized transporter, ASCT2, providing a unique point of intervention.
Okay, fantastic. Now we know the fuel and the transport. That brings us to the massive threat they're fighting off internally. This is where that phrase, living on the edge, really comes into play. Let's talk about ferroptosis.
Ferroptosis. It's a unique and aggressive form of cell death. As you said, it's often described as cellular rust. It's an iron-dependent process driven by the uncontrolled accumulation of toxic lipid peroxidation.
Okay. And these ILC2s, they need to expand rapidly to drive the allergic response, which means they must quickly build new cellular membranes.
And to build membranes, you need fat.
Right.
They engage in high-fat intake and metabolism, but they're not importing just any fat. They significantly increase the absorption of polyunsaturated fatty acids, or PUFAs.
PUFAs.
Yes. And while PUFAs are essential for membrane structure, they are highly susceptible to oxidation. They are literally the kindling for the ferroptosis fire.
So they're importing the fuel for their own massive expansion, but that fuel is highly volatile. They're essentially building their homes out of highly flammable material.
That's an excellent analogy. On one side, you have the danger. They're upregulating proferroptotic genes, absorbing these oxidizable PUFAs. But at the same time, their defense system kicks in hard.
This is where GPX4 comes in.
Exactly. They massively upregulate those powerful anti-ferroptotic systems, glutathione peroxidase 4, GPX4, and the related system thyrodoxine reductase 1, TXNRD1. They have to do this just to survive their own strategy.
And what's the evidence that this defense mechanism is actually winning?
It's remarkably effective. Despite the high uptake of these dangerous PUFAs, the activated ILC2s actually show reduced signs of toxic lipid peroxidation compared to their naive counterparts.
So the defense systems are operating at peak performance.
At peak performance. And this isn't just a mouse finding. Human ILC2s activated by inflammatory signals also showed elevated GPX4 and TXNRD1, confirming this is ramped up in human disease too.
But here's the vulnerability, right? If they rely so heavily on GPX4 to neutralize these internal fat bombs, what happens when you block that single enzyme?
You hit their life insurance policy. In the lab, treating ILC2s with the specific GPX4 inhibitor, RSL3, caused rapid concentration-dependent cell death.
And you're sure it was ferroptosis?
Yes, because it could be entirely stopped by specific ferroptosis inhibitors like ferrostat-1, but not by inhibitors that block standard cell suicide.
And the really critical point is the specificity of the dependence, isn't it? Are the activated pathogenic ILC2s more dependent than other cells?
Yes, that's key. Pathogenic ILC2s were shown to be more sensitive to RSL3 than naive ILC2s. This confirms that this whole tolerance mechanism, it's an intrinsic, crucial requirement for their pathogenic expansion.
Okay.
Furthermore, the source showed they barely use other known antiferroptotic pathways. The GPX4 TXNRD1 pathway is their dominant defense.
So if they are so dependent on GPX4, why don't we just use RSL3, the inhibitor, as a drug?
That's a really important safety concern. While it kills the pathogenic ILC2s, GPX4 is also essential for the survival of many other structural cells, including the epithelial cells lining the lung.
So you'd be causing widespread damage.
Widespread ferroptosis in the lung tissue itself has been linked to, asthma exacerbation and acute lung injury. So blocking GPX4 broadly is just too risky. We need a more targeted surgical approach.
Which means we need to target the non-redundant parts of the ILC2 defense system.
Exactly.
Let's consolidate segment 2 before we move to the therapeutic application.
Quick summary, part 2. The ferroptosis tolerance. The expansion of pathogenic ILC2s requires them to tolerate the high oxidative stress from taking in PUFAs. They do this by massively upregulating the antiferroptotic systems GPX4 and TXNRD1. This tolerance is an intrinsic feature of their pathogenicity, and blocking GPX4 causes immediate lethal ferroptosis.
Okay, this brings us to the therapeutic opportunity. If directly blocking GPX4 is too dangerous, the focus shifts to the partner enzyme, TXNRD1. How does targeting TXNRD1 offer a safer path?
TXNRD1 is an excellent target. It supports the whole system that not only helps make the vital GSH we talked about earlier, but it also independently acts to balance intracellular ROS.
So by inhibiting it, you constrain the entire network ILC2s are so dependent on.
Precisely.
The researchers first proved this using genetics, which is always powerful. What happened when they removed the GPI4 gene specifically from the ILC2s in mice?
The results were definitive. They used a clever model to delete GPI4 just in ILC2s, and when these mice were challenged with an allergen, the ILC2s had a profoundly reduced ability to accumulate in the lungs.
Which means?
It means decreased inflammatory cytokines, dramatically reduced recruitment of eosinophils, and much less mucus in the airways. The asthma pathology was significantly reduced just by removing the ILC2's life insurance policy.
And the elegance here is the specificity. Did removing this defense mechanism affect their normal protective duties?
That is the most exciting point. The genetic deletion had no effect on ILC2 function during a protective helminth infection.
Wow.
This is strong evidence that this metabolic reliance on GPX4 is essential specifically for that aggressive pathological expansion in chronic allergic inflammation, not for their general immune role.
That makes it an ideal specific target. So genetic proof is in place. Now for the practical application, the pharmacological test using the TXNRD1 inhibitor, TRY1.
Right. When the TXNRD1 inhibitor TRY1 was administered intranasally, a very practical delivery method to allergen-challenged mice, the results perfectly mirrored the genetic deletion.
What did it do?
TRY1 effectively ablated ILC2 accumulation in the airways and suppressed IL-5 and IL-13 production. This directly reduced eosinophil recruitment and cellular infiltrates.
And the mechanism was confirmed to be exactly what they predicted, triggering cellular stress.
Yes. The drug worked by causing an increase in lipid peroxidation and reactive oxygen species inside the ILC2s. It just constrained their ability to acquire and metabolize lipids, forcing them into ferroptosis.
And they double-checked this.
They did. They confirmed it by genetically deleting TXNRD1 in ILC2s, which produced the exact same results as the drug, sealing the case for TXNRD1's crucial role.
Let's synthesize this therapeutic breakthrough then.
Quick summary, part 3. Targeting the vulnerability. The GPX4 and TXNRD1 systems are an intrinsic required feature of pathogenic type 2 immunity. Targeting TXNRD1 specifically, as shown by both genetic deletion and the drug TR1, prevents asthma symptoms in vivo without impacting the ILC2's protective roles. Pharmacological inhibition of TXNRD1 offers a promising, highly targeted strategy.
This research fundamentally shifts our understanding of chronic allergic inflammation. We're not just fighting runaway messaging. We're fighting a cell that has adapted to eat its way through inflammation, but whose very survival strategy is its undoing.
It provides a sophisticated link between lipid metabolism and redox balancing required to sustain these pathogenic immune cells. And it suggests that, any tissue-resident immune cell that's activated to rapid proliferation might rely on a similar ferroptosis tolerance mechanism.
So this could apply to many chronic inflammatory conditions.
It's a framework that could, yes.
And it points to potential complementary strategies, not just drug development. The sources discussion section noted something very interesting. A higher abundance of cystine in humans has previously been associated with a higher probability of asthma.
Right.
And now we know that the pathogenic cells rely on this specific amino acid, acquired via that ASCT2 transporter, just to survive.
That connection is fascinating. If the environment is rich in their specific fuel, it enables them to expand pathologically.
So here is the provocative thought we leave you with. Given the pathogenic ILC2s use this specific ASCT2 transporter to get cystine, and given this link between higher cystine and asthma prevalence could, regulating the availability of this amino acid, cystine, perhaps through diet or blocking ASCT2, offer a novel preventive or complementary approach to managing these diseases, working in tandem with new drugs like TI1.
It seems the immune system's specialized hunger might just be the key to turning off its aggressive behavior.
A powerful new direction for personalized medicine and asthma research indeed. We hope you enjoyed this deep dive into immune cell metabolism and the fight against ferroptosis. We'll see you next time.